Milligen, fantastic.
Yeah. Well, thank you very much for inviting me. And I'm sorry I couldn't be with you. We're on the process of extensive fundraising, but I'm glad that I was able to make it finally. I would like to present to you Milligen, which is 27 years in R&D making. And this is about diagnostic. I think all what you are familiar with is mammography, colonoscopy, CT scan, and PSA. And each one of those has a really tremendous limitation as far as its application. For the last 27 years, we focused on identifying secreted protein in the blood that could be indicative for the early detection. And I'm going to share with you just a piece of it. We focused on the 4 major cancers that account for 50% of all cancer, basically. And if we just focus on breast cancer per se, 50% of women for whom the breast cancer sensitivity is like flipping a coin, it's 50%. And a lot of benign conditions are considered positive. When women are considered positive, 95% of them are called back based on mammography diagnosis, and 95% turn out to be false positive after biopsy. So this is the best standard for breast cancer. So we have been, over the last 27 years, trying to identify biomarker for population-wide screening test for early detection, not only for breast, but for colorectal, lung, prostate, and ovary. I'm going to just share with you, since we have just 15 minutes, I'm going to share with you just the data for breast cancer. This is data, it's blind primarily for the early detection of breast cancer. And I would like to share with you this third validation where you could see that the test that we have developed actually identify breast cancer patients at stage 1 and stage 2 at about 90% sensitivity, which is absolutely unheard of. And it recognizes all the different subtype of breast cancer, whether the lobular or adenocarcinoma or other subcategory. So the test is developed primarily for asymptomatic average risk women over 45 years. We presented the data to the FDA and we agreed with them on the inclusion and exclusion criteria in a pre-market application. And we are raising funds as we speak and seeking partnership to move this process forward. This is one application, which is the early detection. In the meantime, we also conducted additional study for the active surveillance of individuals who have had cancer and for the therapy efficacy monitoring. The problem of cancer, it's threefold actually. There's no really sensitive test, easy to use, non-invasive for the early detection. That's number one. Number two, once individuals have been discovered with cancer, they are left at permanent risk of developing metastasis, which would kill them, literally. And three, if they are going through medication, they don't know, and their physician either, whether the medication is working efficiently or not. So we applied the test to show that not only these tests are good for the early detection at 90% sensitivity for stage 1 and stage 2, but we are also able to show and to demonstrate in a longitudinal study that these tests can also have additional intended use. So I'm going to share with you number one, the test for active surveillance. Actually, when an individual is discovered at late stage with cancer, when cancer lodges into metastasis, it would cost between $100,000 and $300,000 per occurrence to take care of that person. There are actually two tests that are available, and some of you may have heard about them, the CA15 and CEA, the only two products available and approved by the FDA. and they both have the same problem. Number one, they only detect individuals when they are at metastatic stages. Needless to say, they are relevant. And number two, they only detect about 25% of the individuals, which means 75% of the rest, although at metastatic stages, it doesn't work for them. So we extended the clinical utility for this product. And for the 4 major cancer, we are basically covering or providing a solution for about 10.2 million cancer survivor between the 4 major cancer. I'm going to share with you just few slides for the clinical study that we have done for active surveillance of cancer of recurrence. So in this case, for example, in these patients, you could see that the CA15, which is the marker that it is being used clinically very, very frequently. Some of the patients use this up to 37 times per year, which means almost literally once every couple of weeks. And you could see that it is really flat all the way down, while our test for the same type of samples of the same patient, you could see that it goes up and it goes down, basically reflecting the clinical status of the patient. We have been able to follow this patient for 12 consecutive years. And you could see on the top of the slide on the right, DF, which means disease-free, when this patient has been declared after all the treatment by the clinician that this patient is disease-free. Indeed, the biomarker or the test indicated that this individual has really very, very low level of the cancer marker. We're gonna see another patient. The patient number 2 is a patient that has been followed for about 5 years. And again, one more time, you can see the CA15, which is a really quite low, almost at the abscess level, while you could see the Milligen test, how it reflects with ups and downs each time the patient reacts to the medication or it starts having metastasis, which is indicated by the M, how the biomarker and the test goes up. The third case, which is a case study, we followed this patient again for about 6 years. And when the patient has been discovered after all these follow-up and treatment as becoming disease-free, no evidence of disease, that's what the NED means. Indeed, we could follow that patient and we could follow that the mammo check, the test, it's following the biomarker in the blood, going down to a level that became close to normal. So in this clinical capacity, we have been able to identify basically that this biomarker can follow the patient and can tell us exactly years ahead of time whether the cancer is recurrent or not. The other study or the other application of this biomarker is what we call therapy response. As I said earlier, once an individual has been submitted to any kind of therapy, whether chemotherapy, radiotherapy, or immunotherapy, there is no way to know whether the therapy is working or not. And we know perfectly well that there is no therapy that works for everybody equally the same. So it is very important to basically monitor the patient as he or she is going under treatment to make sure that there is a positive response to the therapy. And if it does not work, then it is probably much better to stop it and look for another alternative instead of keep providing the same therapy to the patient while the medication is giving side effect. So I'm going to share with you here again an example of 3 different patients. So this patient 1, the ID of the patient is 14,969 on the left side on the top. You could see that this was done within the context of what we call adjuvant therapy. The context of adjuvant therapy, the way it is used, is the patient is submitted to a variety of therapy with the intention to reduce the tumor and then do surgery at the end. So you could see in the beginning, at -8 months, NAT, which basically refer to new adjuvant therapy, that the patient has been receiving a variety of therapy. And when we looked at the biomarker has decreased slightly, and a couple months later, the patient kept receiving the same medication. Nor the patient nor his doctor knows exactly what's going on in the body. But our test was telling us basically that no, after just a couple months, the disease is picking up again, which means another type of the cancer, it started growing, and only if the patient and the physician would have known this, they would probably have done the surgery just 2 months before, after the treatment, not waiting for the entire 8 months. So this is just one case. Another case, so if you follow the first one, then this is one is about the same. The patient has been submitted to new adjuvant therapy, then the biomarker goes down, which means the patient is recovering a little bit, and after 4 months after the treatment, then a new clonal selection of the disease is picking up again. And again, if the physician had known this, he would probably have intervened around 4 months, not waiting for 4 months time, while the patient is kept having the cancer growing up and metastasizing. So the next, it's another case where we are showing again the effect of the therapy. And what this means is you could really, by having this test use it by the patient on every couple of months, you are literally in a position to look at the patients at the physiological and clinical status as if you are looking at the patient under the microscope. This is absolutely new. Nothing had happened for the last 70 years of this nature. And I am glad that we were able to come up with these applications. We submitted the data to the FDA to apply for the 510K and to make this product available all over the places. And we are in the process of completing the request for the FDA, and we are hoping that very soon, within the next few months, we will submit the 510K for the test that we have developed. In the meantime, we have a CLIA laboratory where we are offering these tests for the early detection of recurrence and the monitoring of therapy response, both for breast cancer and for colorectal cancer. The Mammo Check is the test for breast cancer. We have 4.2 million breast cancer survivors in the US, unfortunately, and we have 1.7 colorectal cancer survivor in the US. That's for IvyCheck. So the test is a really protein-based. It doesn't have algorithm. It works yes or no, the same way the PSA test works, the same way the pregnancy assay works. So it's a protein-based one biomarker secreted in the blood. Previously, we have done a study to compare.
Monsef, you got about 5 minutes.
Okay.
Yep.
So we can jump this slide. We have 2 on the market. We have 3 more coming for prostate, for lung, and for ovary. We are raising fund, and we are discussing with, we have 2 ongoing and advanced discussion with 2 major diagnostic company. And we had signed the agreement with a major pharmaceutical company in Mexico. So the test will be available within the next couple of weeks as well.
So who pays for these diagnostics? Is it insurance or who pays for the diagnostics? Insurance?
For now, no, we did not apply yet. We are in the process. We are putting the file together to apply for a new CPT code.
Right, and you're raising 20. How much are you raising for the Mammo Check?
We are raising. I can give you a net shelf. We are raising $70 million for 2 years, to hire 100 sales force and to complete the CLIA laboratory here for manufacturing with 10 additional scientists and 3 additional management team. So when you calculate
can't get you can't get a distributor or something like that.
Well, we looked into a variety of aspects about distribution versus really having our own sales force. And our intention is to educate the market as soon as possible. And obviously, our intention is to have a merger and acquisition down the road, not too long in the future, to allow these products to be available to everybody. Our intention all along and from the beginning was to have a product available, not to do it in a piecemeal way.
So is your intellectual property, it's all global in nature?
You need to talk to our intellectual property guy who has been with the company for the last 27 years. I think he will give you probably a much better answer than I do. Any other question?
So I the reason why I asked is because, you know, that the Middle East tends to fund a lot of these sorts of tests on breast cancer, since the rate of breast cancer is much higher in the Middle East than the United States. So like Saudi Arabia, I think it's 50% higher than the United States. So just food for thought. If you have any, we have some contacts in the Middle East, but you know, if you have some, you might want to reach out to them.
Well, what you are saying, it's true. Nevertheless, we are here 300 million people. In Europe, it's 470 million people. Yes, in the Middle East, the ratio is higher, but it is a small population. And as I said, the IP is really changed from one country to another. And this is why I think the best way you would like to have more information is to talk to our IP lawyer.
Well, I was just talking in terms of raising capital. So anyway, well, listen, this is really fantastic. Any other comments that you want to make or any questions from the audience?
Yeah, this is Edward speaking. I'm happy to provide, if the audience has any questions, I can put you in front of Dr. David Teese, the co-founder that Monsef is referring to. Thank you very much, Monsef. Let's allow room for our other speaker, Dr. John Crisell. Dr. Crisell earned his doctorate in chemistry from UC Berkeley. And
well, I'm sorry, one second. Fantastic job, Monsef. Thanks so much.
Sure. Thank you very much. Take care.
Okay, let's move on to Josh. Josh, you've got 15 minutes.
Hey, thank you very much. Appreciate the ability to speak with you. I must apologize for my attire, amongst other things. I'm laid up with a severely pinched sciatic nerve, so I normally wouldn't be presenting by Zoom or dressed this way, so I apologize for that. I'm going to share my screen and just dive right in here, if I may. Is that showing up? First slide.
Looks great.
Pharma intro. Okay,
yep.
Terrific.
BioQ intro Q2 23. Yep.
So BioQ Pharma, we're based.
You don't see it.
Very good.
Well, we need the tech person then. Let me see what I can do here.
You can't see it.
Just 1 second.
I can see it actually.
I can see it.
We have a presentation that needs to be up on the wall here, and it's not. It's being shared, so maybe it needs to be. Maybe this screen needs to be unpinned or something. I don't know. Yeah, I think you just need to take mine off. That's mine on the right. And take his screen. Yeah. Just give it a second here so we can see it. Again, don't worry, Josh. You've got your full 15 minutes.
Yeah, no worries. Thank you. And you can hear me okay?
We hear you great.
Okay, terrific. It seems that people on the Zoom can see the.
Yeah, maybe it'll catch up.
I think it's just maybe in your.
Oh no! Yeah, this is messed up. Hold on a second here. I'm going to close this. And do I have Zoom? Yeah. Hey, Josh. Josh, we
Yes.
don't.
Yes, sir.
So we don't lose your entire presentation time. Can you just start presenting and go
Yeah,
through the deck?
I'll
Yeah.
start going.
We don't see it here, but
when you guys
we're getting a
get
tech
there,
guy to
you get
address it.
Yeah, no problem. So again, my name is Josh, and our company is BioQ Pharma. We're headquartered in San Francisco, California. We operate in the infusion space. And as you may know, infusions are really defined as the controlled administration of a liquid drug over time into the body. When you think about infusions, you can think about things like chemotherapy, antibiotics, anesthesia, pain management, different kinds of biologic products. And when we started the business, we identified at the macro level several problems and unmet needs in the infusion space. And simply put, those problems are cost, complexity, medication errors, wrong medication given to a patient or in the wrong dose, and breaches in sterility. Bottom line, too many hands touching the drug en route to the patient, and the extent to which the pharmacy, for example, in the hospital, oftentimes being a bottleneck in the patient getting his or her infusion. And so consequently, what we have developed at BioQ with a very proprietary platform is a way to somehow combine the drug and the pump together in a more self-contained package, thereby reducing the complexity of infusions, reducing the cost, and simplifying the infusion process from end to end. So we are a commercial stage company. We have 4 approved products with 44 marketing authorizations around the world. A marketing authorization, for those of you not in the pharma business, means basically an approval, an FDA approval, for example. And typically we focus on hard to prepare or hard to administer infusible drugs. We own all the IP. We've developed everything in-house. We manage the full drug and device supply chains internally. As I said, the platform is very proprietary. We have 54 issued US patents. We also have patents issued in Japan, China, India, the UAE, Saudi Arabia, Europe, Australia, and Canada. We've raised over $100 million from institutional, strategic, and private investors. And in general, I would say our business model and plan is very de-risked relative to the biopharma industry. And what I mean by that is we're effectively taking already approved drugs, essentially just putting them in a new package, but not opening up new questions of safety or efficacy. So yes, our products are drugs because they are drug device combinations and they are approved as drugs, but we are not taking clinical or big regulatory risks, vis-a-vis new drug discovery, et cetera. As I'll tell you about quickly, we also are in the midst of completing a rather compelling, in our view, acquisition of a US FDA approved drug device combination product that's very synergistic with what we do. So again, what we have with BioQ and our Invenius platform is what I liken to a pharmacy in a box that's also a delivery system. So again, it's a drug and a pump together in a single package. In that regard, we're affecting the 3 key phases of the infusion process: preparation, administration, ambulation, thereby lowering the total cost. And total is really the key word. And of course, it differs by therapy and drug and site of care. But in general, we're never bringing a drug on our platform if we can't be convinced at the end of the day, we're going to save the provider some level of systematic total cost. Now, this video I like to show as inspiration for where we started and why we started. This is a anesthetist or anesthesiologist, as you may prefer to call him, getting ready to prepare a certain kind of infusion. So this is a very specific type of infusion. He is about to begin filling this elastomeric balloon pump you'll see on the right. I've seen physicians fill the wrong drug in the pump by accident before. I once saw a nurse doing this at the periphery of the operating theater. She dropped one of the metal caps into the bowl, fished it out with her hand, and kept on filling the pump. So hardly sterile or aseptic. Meanwhile, this is our original first generation product. And he says, I can do all this in 5 seconds. It's a drug and a pump in a single package with a 3-year shelf life. No need for filling, no need for preparation. Just turn the release cap and it's ready to go. So, one specific example, but 24 seconds for the old product on the left, 5 seconds for our product on the right. And as you'll see later, a big delta in overall cost. And as we matured as a company, we got, of course, more sophisticated about the types of infusions we were looking at and focusing on. But our value proposition pillars always remain the same. So number one, everything's safer because it's pre-filled at the factory under GMP conditions, shows up at the provider completely ready to use. Everything's sterile. Value is the most important pillar by trying to lower the total cost. And finally, convenience. While providers don't want to pay for it, everybody loves convenience. And so we have a suite of commercial products in pain and anesthesia, and also a pipeline in antibiotics, oncology, and biologics. Meanwhile, I should point out that our original products you see here on the top were all single-use and fully disposable. So certain advantages in certain sites of care, and particularly in the continuum from hospital to home, but definitely disadvantages in terms of business models. So we have developed this, what we call Invenius One system, that has a universal reusable pump head, as you see on the left, and a single-use pre-filled cassette with the drug and diluent as applicable in the middle. And so at the end of the infusion, the pump head is reused, the cassette is disposed of. Obvious environmental benefit, but also a business model benefit because we rent the pump head to the provider while we sell them the cassette with the drug. So a lot of times people, when they're first seeing our business, think, oh, you're partnering with drug companies to bring drugs on your platform. No, it's the opposite. These are generic drugs. We own the drug. We get the drugs made to our specifications through our contract manufacturers. All of our contract manufacturers at the moment are in Europe, in Germany, and Italy. We get the device made to our specifications. We design and manage that whole process from sourcing the active ingredient through to the batch release. And then we sell through, in certain cases, pharmaceutical companies. You see some here, names that undoubtedly you recognize, or in other cases, in certain markets, through our own direct field force. So the Sandoses of the world, so to speak, are distributors. They're not our suppliers. We're supplying these companies with the fully finished, fully filled drug device combination they're selling on our behalf. Meanwhile, we take a very global approach, and it's, I would say, not unlike some investment templates, in the sense that we're trying to take a lot of shots on goal, make a lot of bets across different therapeutic areas, across different markets. So it's really hedging our bets by drug and by territory. And we are a very international facing company and an unabashed globalist, which some people like and some people don't, but we were certainly not an America first company. And obviously the value creation comes from parlaying the platform across those different drug products. Our focus to date has been entirely on bringing generics onto the platform. That said, it was always part of our plan to ultimately do a branded deal where we do partner with a pharmaceutical company for their branded drug. We've been in the midst of one of those discussions at a very deep level now for the better part of a year. And that really is, changing zip codes for us. It's like moving from where I live in the Mission to Pacific Heights. So it's a much richer zip code, but our thesis was that we always had to show big pharma and biotech that we could develop, manufacture, and register and commercialize these kinds of drug device combinations with generic APIs before we could ever move into the branded game. So just to comment quickly on the competitive landscape, of course, there's all kinds of therapeutic competition. Those innovators that essentially reformulate infusions to injections can be very sexy, very difficult to do, very capital intensive, lots of regulatory and clinical risk. Obviously, there are all sorts of different kinds of pumps that may address certain challenges in the infusion of the drug, the administration of the drug, but not the preparation. Likewise, there are so-called ready-to-use bags that maybe address certain challenges in preparation of the drug, but not administration. So we really combine those two middle themes together in one. we don't do injectors or small subcutaneous injections. So at the moment, we don't see anyone that does what BioQ does. And a lot of times people say, well, this is really kind of an obvious solution to an unmet need. Why haven't the Baxters, et cetera, of the world done it before? At least 4 ideas about that and barriers to competitors entering. Number 1, our patent protection that goes out a long way, and which we're extending all the time. Number 2, I would say the merger of a medical device and a pharmaceutical supply chain is almost an unnatural act. We have a lot of battle wounds to show for it. We've spent, arguably a fair amount of money and a fair amount of time learning how to do that and do it properly. And now we have a recipe book for how we do it going forward. So we're sort of the beneficiaries of our own tough lessons over time. But anyone who has to, wants to do this needs to learn those same lessons, presumably. Obviously,
Josh, we have a few minutes left. Josh, just so you know, we have a few minutes left.
Okay.
About 2.
And finally, the cultural barriers. So, bottom line, drug companies want to make new drugs or have generics, and medical device companies want to make new medical devices, but none of these cultures typically mix. And obviously that's what we've done good at our culture. So just very quickly, let me say, showing you this one that you saw in the video. This is our product for post-surgical pain management. Bottom line, cheaper, safer, more efficient. And just skipping ahead quickly, this is another example in chemotherapy where the reimbursement doesn't change using our system. But the provider is actually getting their savings based on a reduction in need for labor, which is paid hourly in terms of farm tech time and consumables, and has the opportunity to make a bigger bottom line profit. And so again, our focus is on the pharmacoeconomics of infusions and improving that through our highly differentiated proprietary products. And so
how much are you raising? What's the economics and how much have you raised?
Yeah, so just getting there right now. We've raised to date 70 million in preferred stock, 40 million in a convertible note, and we've gotten 31 million in commitments on our Series E round, which we haven't done a closing on yet, but we're staring down the barrel of. For smaller investors, there's still an opportunity to get in on our convertible note. It's not capped. It has a 35% discount to the Series E round. I won't give out valuation metrics, but it is obviously a buyer's market. The valuation, I think, arguably people have found very attractive. Those investors who have signed non-disclosure agreements. The term sheet that we have for the Series E has a quite an attractive pre-money valuation for the E.
In ballpark, can you give that?
It's high double digits.
Okay, great.
And just a quick note on our board. Our board, very seasoned executives from the life science industry. Marian Deckers was CEO of Bayer and Thermo Fisher. Mark Ellert was a corporate officer at Baxter Hospira. Bill Jasper took Dolby public. Lou and Richard, longtime life science executives, and Daniel Tassé is a combination products guy, most recently sold iCarrier to Mallinckrodt for $2.3 billion.
Fantastic. Excellent, Josh. Are there any questions from the audience? Anything online?